All right. Hi, everyone. I'm Veronica Porubsky. I'm a PhD student in Herbert Sarr's lab at the University of Washington. And today I'm going to be talking about representing neurological systems using graph theory and also recurrent neural networks in Python. So basically, there will be two components to this presentation, this tutorial. First, using graph theory to study functional connectivity patterns in neural data. And then the second part will be using recurrent neural network models of neural dynamics to be able to study mechanisms in silico. So I'm going to start with the graph theory portion. I just want to say that both of these projects are in development. So I'll talk about two different packages and those will be released in the near future. But there's a lot of features currently being added. So just a consideration. Okay, so for the first portion of the talk, I'm going to be talking about how you can use graph theory to study functional connectivity patterns, which basically give you an idea of how neural data is correlated in time. So which neurons are firing together. And this can be done in a multitude of ways, but graph theory is one way that we can represent how neurons are correlated. And so I'm going to talk a little bit about how we can then use graph theory. We can build graphs off neural data and then use those graphs to study patterns within the data that might give us an idea of how information is being processed in these true biological neural networks. Okay, the data I'm going to be talking about is going to be calcium imaging data. So dynamic neural activity can be collected using these virally expressed calcium indicators, which essentially are expressed in the surface of neurons so that whenever an action potential is experienced, there's this cognate calcium signal that generates fluorescence over time that can roughly match the underlying electrophysiological activity. So if you want to see a little bit about what the data actually looks like when it's actually being recorded with a miniscope that's implanted in the brain of a live and behaving mouse, this video is showing that. So you can see these little flickers of light, hopefully, in the field of view. This field of view corresponds to this device called a miniscope that's implanted inside of a mouse brain. And in this case, it's recording from a portion of the brain called the dentate gyrus, which is a part of the hippocampus, and it's involved in memory encoding. So once these data are processed from the video, you'll basically get frames that look like this, where you can run a cell segmentation algorithm to identify each of the individual neurons. So that's what these circle objects are. Each of these is a neuron, and then the red and the yellow and white are all different categories of neurons that have been identified in post-processing. But this is basically what you see when you're recording from the mouse brain. And then to further orient you to the way this is performed, you're expressing this calcium indicator. In this case, they're using GCAMP6F, which is a fluorescent indicator that's very commonly used for calcium imaging. And it's expressed in the dentate gyrus. And so the miniscope is implanted above the dentate gyrus, and it's getting a portion of that brain in the field of view. So it's only getting one plane of imaging, essentially, that's collecting. And all the data that I'll be using for this talk was collected by Eric Zhang and Dr. Sean Piantadosi from the Brucus lab at the University of Washington. Okay. So once we've collected these data, when I receive the data, it's been processed into time series data. So you can think of it as basically an array where each of the rows correspond to a single neuron, and you're getting fluorescence data. That data I'm going to then process into graphs, and graphs are, if you are unfamiliar with them, can be roughly summarized as collections of related objects between nodes, collections of related objects called nodes that describe the pairwise interactions called edges between each node. So here you kind of get a very simplistic diagram of a graph where you have two nodes here denoted by these circles, and then an edge between them, which is telling you that there's some relationship that's been identified between the two nodes. And this relationship could be highly variable based on the data you're using. for the data I'm using, the edge is actually going to be a correlation coefficient value. So we'll be using the Pearson's correlation, and that's basically just saying these two nodes have an edge between them when they are correlated in time. So when we construct these graphs, we can then use graph theory to identify topological structure in the networks. So graph theory can help us identify things like hubs, which would be neurons that are highly correlated with many other neurons in the network. It can tell us about the clustering coefficient of each of the neurons, which basically gives us a measure of how a neuron which is connected to neighbors, how its neighbors are also connected to each other. And then communities, which basically give us a even broader picture of the network, where it's creating these larger clusters of within the network of nodes that are all highly correlated with each other. So the data that I'm using when I construct these networks, as I mentioned, it is calcium fluorescence data. So a single event in a calcium fluorescence trace is going to look like this plot on the left. So this tells you about the decay kinetics of a single calcium fluorescent event that's occurring. You can imagine underlying this event, you get an action potential, and that's when you get the change in calcium over time. And that corresponds to this fluorescent signal. And when you take all of the neurons that you're recording from in the field of view, then you can get several neuron time series here on the right, where each of these time series corresponds to a single neuron, and the fluorescence events that are happening over the time series look like roughly like this. And these are just kind of condensed on the time dimension here. So once all those data are processed and we have a series of neurons from the network that we were recording from, we can create our graph. So just as a review, the node is going to be these individual fluorescence time series. The edge then that we're constructing in our graph is going to be the Pearson's correlation coefficient weight between two time series. And there will be a threshold that's applied to binarize the network, so that when you have a correlation above a certain value, so here R greater than 0.2 as an example, then you will add an edge to the network. And the graphs here would be the collection of all of these nodes.
I didn't get that. Could you try again?
Sorry, I activated an assistant. If anyone else didn't get that, please let me know and I'll re-explain. But yeah, the graph is going to be all of these nodes and edges connected to each other. So to kind of give a visual for that will hopefully help explain what's happening here. The graphs are going to be constructed by finding these highly correlated neural pairs. So let's say we have all of these time series in the left hand portion of this diagram, and I've marked 3 neurons, 1, 2, and 3 here, which are colored orange, that are going to be highly correlated. And these orange triangles beneath the 2 time series for neurons 2 and 3 are showing where that time series is highly correlated in time with neuron 1. Okay, so we take all of these time series, we compute the correlation coefficients between all the pairs in this network, and then we get a correlation matrix here, where each of the entries in the matrix is showing you the Pearson's correlation between the pair. And so here you can see that neuron 1 is highly correlated with neuron 2, and then also 3 that were denoted over here. And then all of these data are going to be processed using the threshold, such that those pairs which have a correlation above the threshold value are going to be included in the graph that's constructed. So let's say here you generate an example graph based off these data, and we have neurons 1, 2, and 3 here highly correlated with each other, so they're added to the graph with edges.
I have a quick question.
Yep.
Correlation. Does that mean fires at exactly the same time, or has a similar pattern that could be shifted in time?
For this case, the correlation it's not considering a time shift.
Okay.
That's definitely something I do want to include in a future version of the package, because it could be really important. But right now it's essentially the events are happening at roughly the same time, or
Okay.
pretty close. Yeah. But definitely that's an important feature to add. Okay, so once you have the graph, then you can compute graph theory metrics on it, like the clustering coefficient. You could compute maybe for different portions of the network. So here you get a clustering coefficient of 1, which is the maximum value you can get for a clustering coefficient. It means that a neuron that's functionally connected to a set of other neurons, that those neurons are also connected to each other. In this case, the clustering coefficient for this neuron is 0 because all the neurons it's connected to are not connected to each other. So that's the lowest clustering coefficient you can get.
I have one other question
Yep.
about that. Sorry. When you are correlating things, is it just like an off-the-shelf algorithm somewhere? Because I was wondering, like, the difference between trace 1 and trace 2. One of the differences is that trace 1 has a lot fewer peaks. So if you were just like correlating 1 with respect to 2, that might be different from the 2 with respect to 1, or does the algorithm just sort of include both? Or like, does it look at all the peaks in both traces and sees which ones match, or does it look for, I can imagine something that has like 50 million peaks and another one that has 10, and all 10 of the are matched in the 50 million, but it's sort of just because they're all there.
So
like
it's
this,
computing
yeah.
the correlation across the whole time series. So, which is why a lot of the thresholds you're going to see are going to be relatively low, because actually the correlations, if like when you consider it across the full time series, that's going to kind of average out. If there are periods which are really very inactive in one neuron, but the other neuron has much more activity, you're probably still going to get a relatively low correlation between those two, because the inactive periods are.
So it's correlating the inactive periods as well as the active periods, I guess.
It's
So
correlating the full, so it's a continuous signal that's
okay. So it's not just saying like I have peaks at these places. It's like I have peaks at these places and low at these other places, and we'll see if everything correlates. Okay, cool. Thanks.
Okay, so once you've constructed these graphs, one other thing that I want to mention, which is going to be included in the package, is that there may be some amount of pre-processing steps you want to take on your data set to be sure that it's suitable for this sort of analysis. And probably the most important one is to make sure you're setting a threshold that is meaningful, so that the correlations you're observing, you have some confidence that they're truly like interesting correlations and not just the result of random correlations in your data. And so to do that, to set a threshold or to recommend a threshold based off of the data that's provided to this analysis, there's going to be a shuffle comparison to a shuffle distribution to be able to determine what threshold is relevant for your data. So to create a shuffle distribution of the neural data, you have a time series here, and again, you can see these calcium events happening across the time series. This time series is going to get split up based off the identified onsets of the events. So that's where you see each of these dotted lines roughly. And then each of these segments that's been identified are going to be shuffled in time randomly. So it's going to generate a new time series, a new shuffle time series, where you can see that this red portion was moved over to the next slot, and then the orange portion was moved to later in the time series again in the shuffle time series. And that's going to happen for all of the segments that have been identified using the onsets of these events. And once you do that for all of the neurons in the population, you get these example traces like you have here, where the ground truth is in orange and the random shuffled trace is in gray. And this is just showing the pairs of each of those where the neurons have been shuffled in time. And from that, you want to construct a correlation matrix for both your ground truth and your randomized or shuffled data. And that gives you, and you can visualize that here, where the ground truth again is in orange and the randomized is in gray underlying that orange. And based off these two distributions, if they are significantly different from one another, then you can kind of tell that there are correlations that are meaningful in your data set, in your true data set. And it's not just the result of these random correlations that exist. And the threshold is being set here by identifying where the outliers in the shuffled data begin in the distribution. So for this case, for this data set, a threshold of 0.2 would be a reasonable threshold, and that means that all the correlations above 0.2 are going to end up included in your graphs that get constructed, and hopefully give you an idea of the correlation structure of your true network. Okay, so based off of these sorts of features I just presented, this calcium graph class, CA graph, is going to basically have three core functionalities, one of which is data assessment, which is going to include something like that shuffle distribution I just talked about. It's basically going to give you an idea of how suitable your data is for analysis with graph theory. Then your graph theory portion of this package is going to actually construct the graphs underlying using this Python package NetworkX, which is very well supported in Python for graph theory related analyses. And it's going to perform any analysis of your graph that you find meaningful for analyzing neuroscience data, for example, computing the clustering coefficient. And then there will also be some amount of plotting and statistical analyses that are most relevant for analyzing calcium imaging data included in this portion. And then finally, there's going to be a visualization portion, which helps to visualize the networks. And it uses both NetworkX and also this Boca library, which provides a more interactive format for the visualization. So I'll show some examples of using this now, I guess. Is the size okay on this notebook for the text?
Yeah, should be fine.
Okay. So the first portion of this actual interactive tutorial will be using this calcium graph class and the related visualization and pre-processing classes. So just again, want to note up at the top that the data I'm using is provided by Eric Zhang and Dr. Champion Tadosi. Okay, so I'll just go through some of the functions that are available from this package. Okay, so first I'm going to go ahead and import my package, or I'm going to import modules from my package, which are this calcium graph class, which is useful for performing the graph theory, visualization module, which has all of the interesting Boca visualizations provided in it, and then also this pre-processing module, which has some, which will perform the threshold setting that I talked about. So we're going to import those three things. They're all going to be really useful. And then this is just for the sake of some visualizations of the data, so that hopefully it'll help out with the sake of the tutorial, but it's not necessarily that useful for your analysis. Okay, so then here I'm just going to go ahead and set data path and file name that I'm going to use for the sake of this tutorial. I'm right now going to use this DG dataset, which includes information. It includes data collected from the dentate gyrus using calcium imaging. And then I'm going to go ahead and just to give you an idea of what the data look like when they're being processed, before they're being processed into the graphs, load and visualize this raw calcium imaging data. So again, this is just for the sake of showing you what the data needs to look like before you put it into the CA graph class. Okay, so I'm just getting the data from a CSV file here, printing some information about the number of neurons and time points, and then visualizing the data using pandas. So here's what the data set looks like. It has 122 neurons in it, 3,600 time points. And in the data set, the first row is going to be a time, is going to tell you the time, each of the time points over which the fluorescence values are recorded. And then the rest of the data set is the individual neurons and their fluorescence values over time. So each of these rows is a single time series that's going to be used to compute those correlations for our graph. And I'm just showing a truncated version of the data set here, but this is pretty representative of the types of data sets you would see in terms of the size of neurons and number of time points. Okay, so then the first step in generating the calcium graph object is going to be setting a threshold value. So here I'm just setting it manually. I'm going to use a threshold value of 0.3. This is really commonly used in literature, so it's maybe a good starting place for those that aren't necessarily sure what threshold is relevant yet. And then I'm going to create this, or I'm going to generate a graph object, or this calcium graph object, CG, here by passing in basically the path to the data and the file name of the data that I'm interested in using. And then also I'm providing a data set ID, which is going to be most useful to the person who's collected the data. But in this case, it's basically giving a single mouse identifier so that the experimenter can map this back to the mouse that it was collected from. And then setting the threshold. So the most important parts of constructing your calcium graph object is the path to the file, and then also the threshold. And in this case, we're using a CSV file again to generate this calcium graph object. Okay, so that's going to generate your objects, name it CG, and then from that, you can use that to do a lot of different graph theory analyses. Here you're just going to get a network graph called CG graph, which is going to actually first generate a graph of your network of your data set using the provided threshold. Okay, so once you do that, you can there'll be some attributes to your data set or some parameters in your data set that are interesting. For example, you can tell the number of neurons or the interval between time points collected in your time series. And then also, if you're interested in checking what subject you're working with, you can use the data ID, which has been saved. Okay, so again, yep, this is just telling you some information about the data set itself, 122 neurons, time interval of 0.1 seconds, and giving you the subject name. Okay, so from that same graph object that we just created, calcium graph object that we just created, CG, we can start to analyze the graph that's been constructed. So we can get the clustering coefficient here, and this will return a list of clustering coefficient values. And if we want, we can visualize those using this functionality from the visualization module, which is plot CDF. So that's going to plot the cumulative distribution function of some distribution that you pass it. And we're going to pass it the clustering coefficient. We're going to pass it a label. And we're going to do the same thing for both the correlated pairs ratio. You'll see this code is just the same. We're just going to plot CDFs for that. And then also the hits values, which gives you an idea of whether or not a node in the network is considered a hub. And we'll visualize those three. So plotting those, you get these CDFs, which are going to look highly variable depending on the data set you're using. But here, it's just telling you how what the neurons clustering coefficients in this network look like. This is the correlated pairs ratio CDF, and then also have the hits values, which is going to look really different from the other ones because you'd expect basically hits values, the very high hits values are indicative of a hub in your network, and you only expect to have a few neurons in the network, which are very highly correlated with many other neurons. So here you have relatively low hits value or relatively low number of high hits values nodes. Okay, so then the next portion would be you can kind of you could potentially parse your data to some extent if you want. When you're generating the calcium graph object, you can pass it a NumPy ND array object, and here I'm basically taking a piece of data, generating a NumPy array using this NumPy gen from text, and then I'm indexing a certain time period within the time series. So for the case of this data set, the time series is split in half, collected in two different environments. So I'm splitting it in half here, taking the later half of the data, and this is going to be context A, so it's in one environment. And then I'm taking the first half of the data, and that's context B. So this is the other environment that the animal is in. And then for both of those, I'm constructing this graph object, and then I can get the clustering coefficient for each of those cases. And I can use the visualization module to look at the histograms of those two distributions, of those two clustering coefficient distributions, to try to compare whether or not the graph looks similar in each of those two contexts. And then I can also look at the CDF, which is just a different way to visualize the same data that's in the histogram data here with these clustering coefficient distributions. And in this case, there's not a significant difference between the two contexts, but potentially in a different experiment, you're going to see that there's going to arise this significant difference in how the two different environments or two different stimuli behave, how the graphs behave in those two in response to those two stimuli. Okay, so the next step is visualizing a static graph. NetworkX provides some very simplistic graph visualization methods, and which can be customized, of course. And so this is just going to be the simplest version of the visualization I can do. So with this CG graph object from earlier in the tutorial, I'm going to use plot graph network, and that's just going to draw this graph network. So it shows all the nodes in the network. It has them labeled currently, but you could turn the labels off if you wanted. And it just gives you a rough outline of what this network looks like, how the nodes are connected to each other. But because it's static, it's not that easy to actually explore this. So I've implemented some additional visualization with this Bokeh package. And so we're going to use viz.interactive network, which is going to give us a Bokeh visualization. And for that, the most important thing to pass is the CA graph object. So I'm passing it this object that's already been initialized with a data set. And then there are also some parameters that you might want to change. So I'm adjusting the size of the nodes using the degree. So you would expect very high degree nodes that are connected with many other nodes to be larger in this plot. And then I'm adjusting the color by the communities. So larger clusters of neurons that are related or connected to each other, you're going to see them in different colors. And then I'm just showing that in the notebook. So that's going to generate this visualization, which is, like I said, interactive. So I can kind of zoom in on the network and look at which specific neurons are actually connected to each other. So here, neuron 1 is connected to 6, it's connected to 20 and 27. And then it's also telling some information that you can add to this interactive graph. So it's going to tell you the degree of all the neurons, the hits value, whether or not it's a hub. So 0 or 1 would be in that category, and then the correlated pairs ratio, and then which community the node is a part of, basically numbered by the number of communities that are identified in your graph. And you can save these plots, and you just have some additional tools for exploring them. If you don't want all this dialogue, you can turn that off. And just gives a better way of looking at these networks and being able and allowing you to investigate them in a little bit more depth. Okay. So notably, when you change the threshold, you're going to get a very different network to visualize. So here the threshold was 0.3, which we set up at top, but we can actually change the threshold parameter in our object. So here I'm doing CG threshold and I'm setting it to 0.2 instead. So this is going to update the threshold across the this object instance. And when I now visualize it, you're going to get a totally different graph. So here you see it's much more highly connected because you're going to get more correlations that are being considered part of the graph. And again, I'm coloring it by degree and I'm coloring it by communities and sizing by degree here. And this kind of emphasizes why it's so important how you choose your threshold, because it's going to completely change what your graph looks like, and that's going to change your understanding of the topology of the network. So we'll talk about some functionalities for changing that in a second. One other functionality for plotting here is just, which can be really useful, is plotting match samples. So here, again, I'm going to plot the same data from before, which are not actually statistically different, but it's mapping all the neurons from one recorded in one environment to the other, because it's being sampled from the same time series. So the neurons are the same over time. And it's just showing how the clustering coefficient changes over time for each of those individual neurons. Okay. So those are some of the basic visualization functionalities, but like I said, there's some pre-processing that's available. Most notable is this using the shuffle distribution to identify a threshold value. So for the first part of this, I'm going to basically generate a shuffle. So to do that, I need two different data sets. The first is the actual time series data, which I'm going to generate here from this BLA data set. So that's collected from the basolateral amygdala. It's a new portion of the brain. And then also event data, which is going to be distributed with the BLA data set. And it essentially is a matrix where it has a time row and it has all the neurons as in the data array as well. But here, each row column index in the neuron rows are going to contain either a 0 or a 1. The zeros mean that there was no event detected at that time point, and the 1 means that it does. And it's essentially detecting the onset of events. Right now, this has to be provided by the user, but there's going to be functionality to actually perform the event identification within the package so that the user doesn't have to provide that. They can just provide their raw calcium imaging traces. And then the shuffle is going to happen here. So we're using that prep module or pre-process module here. And the function is generate event shuffle. So it's going to take all the events and slice up the time series, shuffle them in time, and give you a shuffled version of that data set. So just to visualize what the shuffle looks like, here we have plot shuffle example. So it's plotting the ground truth. So this is the true neural data, and then the shuffled version of that data of that neuron is plotted below it. See, it still looks reasonably like neural data, but just all the events are at different time periods. And when that's done for the whole network, then we can look at the threshold like I talked about in the presentation. So here we're going to do prep.generate threshold, where you pass it your data, you pass it your shuffled data and your event data. And this is going to go ahead and return back a recommended threshold. And here you can also plot the threshold. So this is going to plot the shuffled data and the ground truth. And the identified threshold is going to be plotted in red. So here, you get your shuffle and ground truth in blue, and you see your Pearson's R value threshold is being recommended for this data set. It's slightly less than 0.2. One important thing to note is that because these thresholds are going to be highly variable based on the data that's collected from a given mouse and how many neurons are recorded, usually it's useful to process these in batches with a high N number of mice or whichever organism your calcium imaging data is collected from. And batched analyses are going to be implemented in this package as well, so that the threshold would basically be recommended as like an average across multiple subjects. But right now it's being computed for just one single data set. Okay, so then the next step here is just showing that we can generate a graph using this BLA data set, but here we're setting the threshold based on what was recommended above and visualizing it again. So this is a completely separate network, but there'll be functionality to streamline the threshold detection, automatic detection using the shuffle distribution, and then generating graphs using the recommended threshold rather than a user specified threshold value. Okay, so that's some of the functionality I wanted to talk about. I guess I'll ask if there's questions on anything I talked about in the notebook before I move on. Okay. Then we'll move on to the next part, which is just this quick case study of how this could potentially be used. So the graph theory metric that I'm going to talk about for this example is going to be the clustering coefficient, which basically just gives a measure of how synchronized neurons' neighbors are with one another. And the way that's computed actually in the graph is by computing 2 times the number of triangles a given node makes basically with its neighbors. So if a neuron is connected to 2 nodes and there's a connection between those 2 nodes as well, then you form this triangle, divided by the degree of n times degree of n minus 1. And this is basically going to compute it's like a fraction of how many connections are formed with a neuron's neighbors out of the number of possible connections that could be formed. And so when you have a coefficient value of 1, clustering coefficient of 1, you have a fully connected subgraph, and a connection of a clustering coefficient of 0 means that your neighborhood is very sparse. So the example that I'm going to share is from one of the BLA data sets that I was using in the notebook. But basically here, the behavioral paradigm that's being used is this mouse is freely behaving. It's running around this maze called an elevated 0 maze, which basically has these 2 closed walls and then 2 open walls. And in the closed walls, the mice will tend to be more in like a more normal baseline state. When they're in the open walls, they're a bit more anxious because there's no like guardrails around. It's like not enclosed, it feels exposed. And then while they're behaving in this context, they're either in one of 2 stimulation periods. The first is no stimulations provided. So we're recording neurons for 10 minutes when there's no stimulation. But then in the other case, the mouse gets 10 minutes of this 5 Hertz stimulation of these LC to BLA norepinephrine terminals, which has basically been shown in previous literature to induce an anxiety-like phenotype. And so we're creating graphs for this pre-stimulation period here and for the stimulation period as well. This creates very distinct functional networks, and the correlations that exist in each of those time periods are very different. So once you have the graphs constructed based off those 2 time series, or the segmented time series, then you can look at the clustering coefficient in each of the cases. And so here I'm plotting the clustering coefficient in either the pre-stim period and the stim period, or the pre-stim and the post-stim period, which is a third 10-minute section that I didn't show on the previous slide, and then also the post-stim and the stimulation period. And this is an example from just one mouse, but the graphs are significantly different from one another in terms of the clustering coefficient distribution. So this is computed using the Kolmogorov-Smirnov distance, which gives you an idea of the distance between 2 CDFs of a data set. And then across multiple mice here, each of the data points stacked vertically is a single subject. The clustering coefficient is statistically increased in the 5 Hertz stimulation condition across all of the mice. So that's a significant difference as well. So basically, stimulating these terminals may lead to an increase in the average functional clustering in this elevated 0 maze. So that is a little bit of a case study of how the package would actually be used in a behavioral experiment. But just to talk more about the software functionality that will be included in the first stable release, which is currently under development. There are a subset of graph theory analyses that are available via Network X, and there will be more that are going to be included in this stable version that are currently supported. There will be more plotting and visualization functionality. So I want to kind of cover more of the functionality that would be most useful for neuroscience researchers in this domain to look at their graph theory analyses and get an idea of the network topology. Then support for cell identifiers. So if a neuroscience researcher has already performed some analysis on their data and maybe has identified that a subset of the neurons in their data are correlated with a given behavior, maybe they have some identifier for those neurons, like they are selective for a given environment that the animal is in. As long as those are encoded with integer values, I'm going to provide support for including that in the graph so that could be in the visual. So maybe if you generate if you look at the communities in your network, do those maybe also align with identified groups that were identified outside of the graph theory analysis. I also want to add support for batch analyses so that multiple data sets could be passed with just one function call and iterated over to provide a sort of average on some of these graph theory metrics that would represent the full sample of animals. And then also support for behavioral information. So during many of these, there's going to be behavioral information is simultaneously recorded with the neural data. And I want to be able to parse graphs using time periods that are spent in specified behavior. So again, this behavior would probably be classified with integer values so that I could parse graphs into, let's say that the animal is either in the open portion of the elevated zero maze or the closed portion. I can go and segment the time series based on which behavior it's being spent in, and then use that to generate behavior graphs, which could be interesting. And then also implement a formal sensitivity analysis and some other pre-processing steps to decide whether or not the data set is suitable for graph theory analysis. Any questions on this? Because I'm going to change to the next topic. I'll be kind of quick, I guess. Okay. All right, so that is the CA graph class or package. The next one is going to be a package which is going to be used to generate recurrent neural network models of neural dynamics to study mechanisms in silico. And this is in an earlier stage of development, so I'll kind of talk about some caveats with this. Recurrent neural network models can be used to study neural dynamics. This is, I should have edited this. It could be for the anxiety circuit, which is specific to my research, but actually it could be used for any neural population. And in my case, I'm studying the mechanisms underlying anxiety, but this could be used to study other mechanisms that are interesting. So this is an example of a neural network. It looks roughly like a feed-forward neural network, and the difference, which most are probably familiar with, but the difference between a feed-forward neural network and a recurrent neural network is that you're going to get feedback between layers that helps you process sequential data where you want some memory of the previous information that's been seen. So what are recurrent neural networks? Well, like I said, these neural networks can incorporate feedback within and also between layers in your network. It enables processing of sequential data like time series, which I'm most interested in, and it remembers information from the previous states in the weights of the network. And I'll talk a little bit about why they're actually good for neural data. So in the previous portion of the tutorial, I talked about using graph theory, and graph theory is really interesting for getting a picture, like a static picture of what the network looks like, and understanding the correlation structure within your network. But it looks at it with just like a snapshot in time, and it's not a dynamic model. But recurrent neural networks actually offer you the ability to study neural systems by creating a dynamic model that can actually be simulated and like edited to generate complex dynamics and to test experiments. So they can exhibit these complex dynamics over many spatiotemporal scales. Neural circuits recorded from animal brains have internally generated dynamics, and in order to model those, you do need both feed-forward and feedback mechanisms to control the dynamics, which RNNs have. You also need, I think that neuroscientists need a scalable way to process large and complex data sets, and RNNs can be trained on time course data that's collected from a single subject, a single experiment, and provide this in silico representation of the original data to be used as a sort of virtual patient. So I'm really interested in generating these so that we can run experiments in silico that don't require us to actually experiment in the mouse, and we can get a better confidence in the experiments we do want to run in mice. So how are we going to use trained RNNs? The first would be to propose and test mechanisms for example, how information is processed. to be able to determine like whether there are neuron subpopulations that have an extreme influence over the behavior or emotional state, and then also to guide experimentation and, like I said, increase our confidence in the in vivo experiments we do want to perform. So I'll just go the broad overview of this, what this class will entail when it's released. But there are, again, three main functionalities here. The data assessment, again, which is going to be specific to calcium imaging data. It'll try to give you an idea of whether or not your data is prepared properly for analysis with the RNN. So one example of what might be checked would be a sparsity assessment. So if your data is very sparse, it doesn't have very many events over a time series, that might actually make it more difficult to train the RNNs. And then there might need to be additional functionality included in the RNN to make sure it can accommodate that type of data. So the next portion would be actually the RNN. So constructing a network that is useful for representing your neural data and training it on the experimental data you have available. And then also performing experiments. So maybe knocking out some neurons in the data and evaluating the input on the predicted activity. And then also visualization. So visualizing training steps, visualizing how the weights are distributed in your network. Yeah, I know I'm a little bit short on time, but I'll try to be kind of quick with this. So the RNNs that I'm proposing to use are actually going to be fully connected with units that serve both as input and output units. The example I'll show at the end is not using this format, but essentially the goal is that we'll have neural data that looks just like we talked about in the CA graph class, and each of these neurons is going to correspond to some neuron in the computational neural network that's going to be represented here, such that you train the network on neural data and then use it to reproduce neural data that roughly it looks like the original data it saw, and you basically get a one-to-one mapping from all the in vivo data to the RNN output. So you want there to be a direct match between them. And during training, the main objective that's being trained is to minimize the mean squared error between the predicted data and the original data. And I'm sure you've seen probably a loss function like this before. It's really common. You basically trying to minimize these the residuals between the predicted data and the actual data. And so over time, you'll have multiple iterations of training, and initially you might have a bad fit where this black line is trying to approximate the underlying cyan line, and then over time you're going to get a better match between the in vivo data and the RNN, and that's done by updating these weights in the network here. All the recurrent weights in the network will get trained, and over time you're going to get a difference in the distribution between the original randomized weights and the trained weights. And I guess we'll skip this for the time being. It is pretty much talking about how the update is performed in the RNN, but that's kind of standard in RNN training. And I think I'll break for a moment to go to another short tutorial on that. So
Veronica, I wouldn't worry about going late. We're
Okay.
just recording things anyway, so.
Okay. Well, either way, the break here is probably good because the behavioral stuff is a little bit separate, but so this is, like I said, it's further behind in development, but I wanted to kind of show what the goal is with this project. So with both this and also the graph theory project, the goal is really to make it really easy for neuroscientists, or maybe anyone that doesn't really necessarily have a lot of computational experience, to get these graphs up and running, or get an RNN up and running and be able to actually perform experimentation or computational analyses on these neural data that otherwise would be a little bit too complicated. So the goal with this is really to minimize the amount of specialist hyperparameter setting that needs to be done in order to get an RNN working, but still provide like a really strong model. So that's the goal here. So with this, I'll just run through some code. So there's going to be a package called CARNN, calcium RNN, and from that I'm importing this CARNN class and also some functionality to plot the principal components analysis activity later, and then also importing some plotting functionality. So here I'm going to use the DG data set from before for test data. And this is pretty much what I visualized as the simplest form of getting an RNN up and running without having to provide too much information into what that RNN looks like. So here I'm constructing an object from the CARNN class. I'm passing in the data path. So basically all it's getting is the CSV file that I specified, and I'm creating this CR object. And within that, you'll automatically be able to generate a neural network without much need for additional hyperparameters. And then you can choose to train the model. So here, minimally, you would just need to set the number of epochs, but of course that could also be set within. And then I'm going to, this will basically train the model. So internally it'll update that network state. And then here I'm going to run a prediction. So here, without specifying which data to predict on, it's basically splitting up the original training data into a predicting set. So ideally you're going to probably pass it of a test set, or there will be a test train set constructor within this CARNN class. But here I'm generating some predicted activity and just using the simplest format. So a lot will actually be changed, or there's going to be a lot of functionality built into this that is not there. This is really just like a vision at this point. But right now I'm just going to go ahead and train the internal network and run the prediction. And right now it's using, it's going to basically, for the prediction, it will be essentially automatically be taking a single data point. So it'll take a snapshot of the whole network at time 0, and then it'll try to predict the next point, and it'll iterate over, it'll just move one by one. So it's not actually giving a good prediction, like a super useful prediction here. The more useful would be to train the network, update the weights, and then only give it a snapshot of time 0 and try to predict the full time series, which will be a future iteration of this. But for right now, I'm just trying to give an example. Sorry, the sizing is a little off, but here it's basically giving you, it's plotting a heat map of the true data. So when you see the red or yellow, it's indicating that there's neural activity happening at that time point. And you get a pretty good map between the model prediction and the true test data. I know that's hard to see, so I'm going to also visualize it a couple different ways. But so here is projection of the neural data of the population activity into 2 principal components. And again, between the model and the true data, you're getting pretty good agreement with where the trajectories fall across those 2 principal components. And then just finally, an example of what you would want the time series to look like. There's good alignment between the model and the data in this case. This is kind of this is really a trivial example because it's only predicting one time point at a time, which is reasonably easy to do. But ideally, this is the type of fit you would want to get. And hopefully you could predict from time 0 and get a reasonable approximation of the population dynamics, at least, if not the actual neural time series, which would be a bit more complicated. So that's just a very, very short example of what this will do in the simplest state. In the more complex state, as we move on, there are other features I want to include. For example, I want to be able to add an output unit that can represent behavioral data. So this could basically, in this case, you would get these readout weights that are added to your neural network. So here you have the original recurrent neural network, which is just trained on the neural data, but then you include additional readout weights that map to a classifier, which is telling you, based on the neural data, it sees right now what behavioral state is the animal in. So maybe that could be if it's in the interior or if it's in the closed or open arm of the elevated zero maze. It should be able to predict that based off of the neural data and the readout weights. Yep, this is just going more into what that output unit would look like. Basically, each time point, it would predict a sequence that is a 0 or a 1, maybe for the behavioral state. So it could be closed arm, open arm of the elevated zero maze here. Yep, and that will be, I'll have a sigmoid activation function for binary, which is kind of like an in the weeds detail, binary classification. And then, yep, I'll like kind of showed an example of it, but there'll be dimensionality reduction incorporated within the package to be able to assess the impact of modifying different units of the RNN, so individual neurons in the RNN, and checking the impact on the population activity by using something like principal components analysis, which is just an easier way to visualize how the population activity as a whole changes. And with the first stable release of this, there should be this very simplistic generation of an RNN. So no hyperparameters are necessarily required by the user, but you can still get an RNN up and running with very simple function calls. And then also more flexible RNN generation for those that maybe do have more RNN modeling experience to be able to set the hyperparameters in a more meaningful way, maybe to give them more confidence that the model they're generating is really tailored to their data set. And then I also want to include predetermined experiments. So for example, you could call a function to knock out a subset of neurons that would be, you would just have to provide the neuron identifiers, or to modify the weights between neurons, maybe, or reduce the activity of some of those neurons and basically evaluate the output on the population activity. And then, like I said, I want to incorporate this behavioral data with a sort of classifier network so it can be added on, and it should be kind of flexible so that, well, the data will have to be prepared. So it's either a binary or some integer value base for whether the behavior is in one state or another. But this should be flexible to be able to accommodate multiple different types of behaviors that are being recorded. Yeah, I'll probably talk about some of the functionality available, but in each of these, but ultimately these are going to both be available on PyPy. Planned release dates for those are the end of March for the calcium graph class, and then also the end of April for the calcium RNN class. So there should be good, a pretty robust package available for both of these soon. And then hopefully I can get feedback and start implementing the functionalities that are actually, that folks who actually are using the software are most interested in adding to the package. Yep. Just want to quickly acknowledge both the SARA lab and also the Brucus lab for all the advice on the software, and also for the Brucus lab providing actually the data that I've used to try to inform the analyses that I'm doing and work on the software. So I'm really grateful to both labs. And that's all I have for now.
Clap, clap, clap.
I'm gonna go ahead and stop the recording. I think we lost Jim. I guess any questions on this or comments, feedback?
I have a question. So when you were doing the tutorials, so you mentioned when you do the visualization, you use the network X, right?
Yeah.
So the node can show many details about the information. Is it from network X, or how did you add that?
No, it's from this Bokeh package. I found it to be a really great package for visualizing network X, but it's actually reasonably complicated in how it specifies the graphs. So for my package, I've tried to condense that information as much as possible so that others don't have to deal with the complications of that package. But yeah, it's from Bokeh. It's called Bokeh, B-O-K-E-H.
Okay. So does it have like animations?
I haven't seen that functionality. I haven't used that. But I
Okay.
guess, what do you mean by animations?
Because right now the first part of the work is like kind of a static visualization, but later you said you have a recurrent dynamic network. So I'm wondering if there's animations maybe you can use when you show the recurrent networks dynamics. I don't know if it's possible. It's just just for curiosity. Yeah, but yeah, I just wonder.
Yeah, I'd have to look into it. I'm not sure. I also don't know if I'll use that package for the RNN portion of the project, but like
Mhm.
I haven't thought too much about which visualizations I'm going to provide for the RNN package, so it might be useful, honestly. Yeah.
Because sometimes when I see animations, it's kind of very impressive for like dynamics showing. Yeah.
Yeah, for sure.
Thank you so much for your talk.
Thanks for having me. Thanks for hosting the series and organizing it, June.
Sure. So do you have other questions or comments for this great talk? If not, then I will stop recording.